In our first week lecture, we're going to cover topics such as science, scientific method, basic principles of taxonomy, and basic chemical concepts. So first of all, what is science? So science is not the truth. It is a knowledge that covers the general truth and uses the scientific method. Biology specifically studies life. This life may be of all sorts and kinds, different sizes, from smallest viruses to the entire ecosystems. Ironically, most of the subjects that have a word science in their name cannot be considered, an actual hard science that is based on the scientific method, such as, for example, political science. Natural sciences can be divided into two large categories: physical and life. Physical sciences include astronomy, geology, physics, and chemistry, while life sciences include biology and medicine. Science is based on the application of the scientific method. The first person to document basic principles of the scientific method, Sir Francis Bacon, a British naturalist and basically scientist. And the idea behind the scientific method is that it is used for logical and rational problem solving. It starts with an observation. Based on the observation, the question is being asked, and based on that question, the hypothesis is formed. A hypothesis is a suggested explanation of the event, so to say, an educated guess that is produced based on the observation and the question. Once the hypothesis is formulated, it is tested. It is tested in an experiment, and then the results of that experiment are analyzed. The important feature of scientific hypothesis is that it must be verifiable and falsifiable. What does that mean? If the hypothesis is verifiable, it means that it can be potentially tested, that there is an experiment that can be performed to examine the validity of the hypothesis. Falsifiable hypothesis means that there is a possible outcome of an experiment that potentially can disprove the hypothesis. One of the most famous examples is Michelson-Morley experiment that tested special theory of relativity by Albert Einstein. It was performed at Case Institute of Technology here in Cleveland. There was an outcome that would prove Einstein wrong. However, it never panned out, and the outcome actually confirmed the correctness of the special theory of relativity. If hypothesis is supported, more experiments are usually done to further test and support or falsify the hypothesis. If it's not supported, then different hypothesis is formed, and the experiment is done to either prove or disprove that hypothesis. In the experimental settings, every experiment has a variable. A variable is a part of an experiment that can change, a part of an experiment that can exist in different types or in different amounts. Variables can be dependent or independent. So the dependent variable basically depends on the value of another independent variable. The value of the independent variable is controlled, but the dependent variable is actually one that is being tested. So when you, for example, test how much salt can be dissolved in 1 liter of water, the amount of salt that is dissolved is dependent variable. The amount of water, the temperature of water are independent variables. They are maintained at the same value. When a hypothesis is tested multiple times, it becomes a theory. A theory essentially is the explanation of natural phenomena that is done through multiple tests, through observation, multiple experiments. So, based on the approach, science can be divided into descriptive and hypothesis-based. Descriptive science focuses on the observation, exploration, and discovery, the documentation of the observed natural phenomena. On the other hand, hypothesis-based science initially forms a scientific question, forms scientific hypothesis that is testable and falsifiable to answer this question. So, when scientists explore the world, when they apply scientific method, there are 2 types of scientific reasoning. One of them is an inductive reasoning. In the inductive reasoning, scientists perform multiple observations and then draw general conclusion. So, observations, for example, that led Darwin to formulating his theory of natural selection is that members of specific species are different and they compete for the resources, and the species are generally adapted to the environment. All of those were observations. And the conclusion was that individuals that are adapted to the environment the most are more likely to survive and pass their traits to the next generation. Inductive reasoning is more common in the descriptive science. On the other hand, deductive reasoning draws conclusions from a general premise, and then specific results are predicted. For example, we know that individuals that are most adapted to the environment are more likely to survive. And then we make a statement, we make a hypothesis. If the average temperatures will increase, for instance, due to the global warming, the individuals that are better adapted to warmer temperatures will outcompete those that are less adapted. This is an example of deductive reasoning that is more common in a hypothesis-based science. We have to acknowledge that descriptive science and hypothesis-based science constantly communicate, so to say, with each other. And the conclusions that are made by descriptive science can then be applied by a hypothesis-based science. Science can be divided also into basic and applied. Basic science essentially expands the fundamental human knowledge. Applied science, on the other hand, focuses on solving the specific real-world problems. Human Genome Project, by itself, did not solve any particular problem, but it was immensely useful for solving certain medical problems associated with the genetic disorders, i.e., in other words, basic science was instrumental for the applied science. Since biology focuses on the studies of life, we need to talk about the properties of life. They listed in this table 8 basic properties of life. They're pretty self-explanatory. We will still go over them. So first is the order, which means the organisms have a high level of organization. Second is response to stimuli, which means organisms can either positively or negatively respond to stimuli. Positive response is considered to be towards the stimulus, and the negative is away from the stimulus. Living things can reproduce, which means the genes in the DNA are passed along to the offspring, basic heredity, and offspring belongs to the same species as the parents and will have similar characteristics. Adaptation, meaning that organisms that are better adapted to the environment will survive and reproduce better, so increased reproductive success, increased reproductive potential, and the adaptation is driven by natural selection. And natural selection essentially is the selection based on the changes in the environment, certain selective pressures. Organisms can grow and develop, so genes drive the development of organisms by providing specific instructions. Organisms become larger, organisms often become more complex. Regulation and homeostasis, meaning that organisms tend to maintain stable yet dynamic internal environment. Energy processing, organisms use different sources of energy for the metabolic activities and can convert from one energy source to another. And finally, evolution. Evolution means that organisms evolve, and they evolve by being exposed to various selective pressures. So organisms that are more fit for the environment have greater reproductive success, and it's important to understand that that will be reflected by the changes in the genetic makeup in the population over a period of time. Now, in order to speak professionally about life science, we need to talk about hierarchy of structures in biology. It all starts with an atom, which is a basic unit of matter that cannot be split into smaller bits chemically. Atoms comprise a molecule, and molecules comprise organelles, structures within the cells. Organelles form essentially cells, and different cells form a tissue, such as a connective tissue or nervous tissue or muscle tissue or epithelial tissue. Tissues form organs, and organs form organ systems. For instance, different tissues form a heart. Heart and blood vessels form cardiovascular system. Organ systems form an organism. Organisms, different organisms of the same type form a population. Populations of different organisms that reside in the same location form a community, for example, plant and animal species in the park. Various communities form an ecosystem, and these communities that form an ecosystem. So ecosystem includes not only living organisms, but also abiotic factors such as lakes and soil. That is abiotic factors. All ecosystems on Earth form the biosphere. Now we need to highlight the difference, fundamental difference, between two types of organisms, eukaryotes and prokaryotes. So what is the difference between the two? Eukaryotes are organisms that have a nucleus and the membrane-bound organelles. On the other hand, prokaryotes are single-celled organisms that do not have membrane-bound nucleus and do not have membrane-bound organelles. Probably the most fundamental property of life is that it consists of cells. If a biological structure is not made of cells, it is not considered to be living. For example, viruses aren't living because they lack cellular structure. When we talk about organisms, we need to classify them somehow. The method of classification of living organisms is called a taxonomy. The first taxonomical classification was proposed by Carolus Linnaeus, Swedish scientist, that identified two kingdoms, animals and plants. It was quite a long time ago, as you can see, almost 300 years ago. Later, 100 years later, Ernst Haeckel, the German scientist, added two more kingdoms, protists and Monera. Protists, or protista, included all unicellular eukaryotes, such as simple fungi, unicellular algae, and protozoa. Monera, on the other hand, included all unicellular prokaryotes. In 1969, not 100 years later, Robert Whittaker added fungi as a kingdom, and that was the classic representation of the classification of all living things by 5 kingdoms. It's important to highlight that these classifications were based on the phenotypes, the structures, the morphological and physical traits. Later in 20th century, American scientist Carl Woese suggested a different approach to classify living organisms. He analyzed conserved 16S ribosomal RNA gene in different prokaryotes. In other words, he was looking at the similarity of genotypes. What does it mean that it was conserved? So conserved means that it is essentially doesn't change much over evolutionary time. Conserved genes are relatively resistant to change. Based on his analysis of 16S ribosomal RNA gene in prokaryotes and later 18S ribosomal RNA gene in eukaryotes, Woese reclassified all organisms into 3 domains: bacteria, archaea, and eukarya. If you look at the phylogenetic tree, you can see that all organisms share the last universal common ancestor, or LUCA, and this node shows you that archaea and eukarya share the same ancestor, and bacteria don't. In other words, I usually say that bacteria is like so archaea and eukarya, like brother and sister, and bacteria for them is a cousin. Now, the domain is the highest level of biological organization. When we start going down and break it down further, so from domain we go to a kingdom. We still keep some kingdoms. We keep kingdom Animalia, Plantae, Fungi. Within kingdom there are phyla. For instance, dog belongs to a phylum Chordata. Within phyla there are classes, such as Mammalia. Within classes are the orders, such as Carnivora. Within orders there are families, Canidae. Within families there are genera, or single genus, like Canis, and within genus there's species, Canis lupus, dog or wolf. A fundamental difference between all eukarya versus bacteria and archaea is that archaea and bacteria are always unicellular and prokaryotic, while eukarya, obviously eukaryotic, and can be either uni or multicellular. Now that we discussed the basic properties of life, it is time to talk about chemistry. We're going to start our conversation with the concept of an atom. So, atoms comprise matter. What is matter? Matter is anything that occupies space and has mass. Matter is composed of elements, and elements are different between each other. Each element has its own unique properties. The definition of an element is that it is a pure substance that cannot be chemically transformed into other elements. For instance, oxygen cannot be chemically transformed into nitrogen. Iron cannot be chemically transformed into copper. All elements consist of their specific atoms, atoms that are unique to that element. An atom is essentially the smallest component of an element that keeps all of its chemical properties and cannot be transformed into a different atom chemically. Structurally, atom contains a nucleus, shown here, that is a very, very small nucleus. And nucleus is composed of protons that are positively charged and neutrons that do not have a charge. Nucleus is surrounded by an enormous amount of space that contains electrons, so-called an electron cloud. In terms of the mass, obviously we do not express the mass of those particles, such as an electron, a neutron, or a proton, in grams. We use atomic mass units, or AMU. You can see that compared to the mass of a proton and a neutron, the mass of an electron is very small. So for our intents and purposes, we consider the mass of a proton and neutron to be 1, and the mass of an electron to be 0. As you can see, it's just an approximation that makes our life much easier in terms of the description. And we also use, instead of charges in Coulomb, we use unit charges. So electron has a -1 charge, a proton has a +1 charge, and neutron has a 0 charge. So you can see that pretty much all the mass is concentrated in the nucleus, while most of the volume is concentrated in the electron cloud. So the model of atom that we are using is called a Bohr model. In Bohr model, electrons orbit the nucleus. When they orbit the nucleus, they don't emit any radiation. Radiation in this case is just electromagnetic waves, but they can move to a different orbit if they absorb a photon, a particle of light, or emit a photon. So atoms have different, as you can see, different orbitals for electrons, and the lowest possible energy orbital is the most stable, and it's the closest to a nucleus of an atom. The simplest atom is an atom of a hydrogen. Hydrogen has 1 proton in the nucleus and 1 electron on the first orbital. When electron is in that orbit with the number 1, it has the lowest energy, so-called ground electronic state. Higher orbitals, like the orbital number 2 and orbital number 3, they have greater energy. In order to move the electron to that next orbital, the electron has to absorb a photon. You can see it here. When the electron goes back from that excited state, goes back from the second orbital, for example, to the first, a photon is emitted. In other words, we can show it this way. So in this case, in order to move, the photon has to be absorbed, but when electron moves back, photon is emitted. So as you can see, in order to move electrons from one orbital to another, the external energy is required. What's important to understand is that the amount of energy that is required to move the electron from the ground orbital all the way up to here, whatever, orbital 5 or and the amount of energy that is released when an electron moves back to the ground state. These energies are the same. So elements differ between each other in terms of the number of protons in the nucleus. The number of protons is called an element atomic number, or Z. Essentially, the value of the atomic number determines the identity of the atom. So, for instance, any atom that contains 6 protons is carbon, and its atomic number is 6. Since atom is electrically neutral, the number of protons equals the number of electrons. So that number Z not only tells you the number of protons in the nucleus, but also tells you the number of electrons there. When you combine the number of protons and neutrons in the nucleus, it will give you a mass number. Remember, the mass of electrons is so small that we do not take it into the account. For example, if you look at the atom of helium, it has 2 protons, 2 neutrons, which means its mass number is 4. If you look at the atom of magnesium, it has 12 protons, 12 neutrons, so its mass number is 24 and atomic number is 12. You can also see that the atomic number is written as a subscript to the left of the name of the element, and mass number is written as a superscript to the left of the name of the element. So what is going to happen if the number of protons and the number of electrons aren't the same? So for instance, here in this example, helium lost 2 electrons. It still has 2 protons. It's still helium. It just lost 2 electrons. So this is an ion, positively charged ion called a cation. In the example of magnesium, magnesium also lost 2 electrons. And in this case, magnesium forms an ion, another cation, because the number of electrons is smaller than the number of protons. This ion will be positively charged. If the number of electrons is greater than the number of protons, the ion will be charged negatively and will be called an anion. We're going to go back to this concept later. To summarize what we just talked about, an atomic number is the number of protons, an atomic mass is the number of protons and the number of neutrons. If you know the atomic number and the atomic mass, by subtracting atomic number from the atomic mass, you can find the number of neutrons in the nucleus. We mentioned that the identity of an atom is determined by the atomic number of an element, essentially by the number of protons in the nucleus. But can different nuclei have different number of neutrons? The answer is yes. Isotopes are different forms of an element which have the same number of protons but different number of neutrons and therefore different atomic masses. For instance, magnesium is a mixture of 3 isotopes: magnesium 24, 25, and 26. All of the magnesium atoms have 12 protons in the nucleus, but magnesium 24 has 12 neutrons, magnesium 25 has 13 neutrons, and magnesium 26 has 14. Essentially, mass number demonstrates which isotope of an element we're talking about. Some isotopes are unstable. We call them radioisotopes. Radioisotopes emit radiation when they decay. They are generally unstable. For example, carbon-14 is a radioisotope that naturally occurs in the environment. It is created by cosmic rays in the atmosphere and incorporated into living things. And the relative level, the ratio of carbon-14 in a given organism matches the relative ratio of carbon-14 in the atmosphere. When an organism dies, it does not ingest carbon-14 anymore, and the amount of carbon-14 in the organism goes down. Which means, since we know what is the half-life of carbon-14, it can be used to determine the age of the fossils. Before moving on, we need to mention what half-life is. Half-life is the time that it takes for the half of the radioactive atoms, for instance, atoms of carbon-14 in a sample, to decay, in this case, to decay into nitrogen-14. Scientists can analyze the ratio of carbon-14 in the fossil sample and, knowing its half-life, determine how old, literally how old, the sample is. It's called carbon dating. Okay? The precision of the method isn't particularly great. It's about 50,000 years, but still it will allow to determine whether certain organisms were contemporaries or not. In this table, it is more for your own information, for your own exploration. You can see the isotopes of the light elements, and based on the natural abundance, some of them are more stable, some of them are less stable. Some of them are radioactive, some of them are not. So all the elements are organized in the periodic table, which was developed based on the previous data by Russian chemist Dmitry Mendeleev, shown here in this illustration. Mendeleev essentially recognized that properties of elements are in the periodic relationship. He claimed that he saw this, so this is the original periodic table. He claimed that he saw his periodic table in a dream. Interesting fact about Mendeleev, he was a craftsman. Just as a hobby, he made suitcases. So what's important, not just that he organized elements, the attempts to organize them were known before, but he used the table to predict the existence of elements that were discovered later. For instance, he predicted the existence of gallium, similar to aluminum, and he also predicted, here's gallium, and here's aluminum, and he also predicted the existence of germanium, similar to silicon. Here's germanium. So essentially, he demonstrated that the properties of the elements are periodic function of the atomic numbers. And he arranged elements in the increasing order of the atomic numbers. And atoms with the similar properties were grouped within the same vertical column. So when you look at the composition of the modern periodic table, you can see that there are groups. Those are vertical columns numbered from 1 to 18, and then there are periods, the rows. Elements within the same group, for instance, elements in the group 18, share similar chemical properties. All of these are noble gases. Elements in the group 2, for example, share similar chemical properties, and most of them belong to alkaline earth metals. When you look at the label for a particular element, you can see its symbol, you can see its atomic mass at the bottom, and you can see its atomic number. In this specific table, elements that are beige are metals, for instance, molybdenum. Elements that are pink, metalloids, sitting somewhere between metals and nonmetals, and elements that are light blue are nonmetals. The black symbols represent the solids. Blue symbols represent the liquids. There are only two liquids at the normal temperature, bromine and mercury. And red symbols represent the gas at the normal temperature. Now, we mentioned before that atoms can gain or lose electrons. And when that happens, they form charged particles called ions. Knowing the periodic table, you can predict whether an atom will form a negative charged ion called anion or a positive charged ion called cation. It can also predict the charge of an ion. For instance, main group metals, metals of the first and second group, lose electrons. And when they lose electrons, they end up with the same number of electrons as the preceding noble gas. For instance, when sodium loses an electron, it has the same number of electrons as neon. When rubidium loses an electron, it has the same number of electrons as krypton. So alkali metals that are in the group 1, you can see them here. These are alkali metals. They always lose one electron and form a cation with a single positive charge. Alkaline earth metal from the group 2 lose 2 electrons and form cations with a positive 2 charge. Elements that are nonmetal usually gain electrons. You specifically must know the properties of the elements of the 16th and 17th group. Elements of the 16th group gain 2 electrons, forming anions with a negative 2 charge, and elements of the group 17 gain 1 electron and form anions with a negative 1 charge. But why does that happen? Why do they lose or gain electrons? Well, turns out it's all about stability of an atom. The most stable elements, elements that do not engage into much of the chemical transfigurations, are noble gases. Noble gases in the group 18 are basically inert because they have their outer shell, also known as a valence shell, completely filled. So every atom, so to say, I don't want to make it too anthropomorphic, but every atom kinda tries to end up with the octet, 8 electrons on the outer shell. So any ion has the greatest stability when its electron configuration is the same as the electron configuration of the previous adjacent noble gas. Sometimes afterwards. For instance, sodium has 1 electron on the outer shell, shown here in red. When it loses it, it becomes sodium cation, and it has an electron configuration of neon. Chlorine, on the other hand, has 7 electrons on the outer shell, shown here in red. When it acquires an electron, it has an electron configuration of the next noble gas, adjacent to it, argon. So, octet rule essentially says that atoms are most stable when they have 8 electrons on the outer shell. In order to achieve that octet, atoms can either exchange the electrons when electron goes from one atom to another, or share them. When electrons are changing the ownership, the resulting compounds form ionic bonds. When electrons are shared, they form covalent bonds. Ionic bonds represent the electrostatic attraction between cations and anions. Remember, cations have a positive charge and anions have a negative charge. An example of an ionic compound is sodium chloride. Sodium has 1 electron on the outer shell, which is acquired by chlorine, which has 7. So now we have sodium cation with a charge +1 and chloride anion with a charge -1. Ionic compounds such as sodium chloride usually form crystals. They're usually solid, rigid, and brittle. They have high melting and boiling points and can conduct electricity when they are either dissolved in the water or melted. Ionic compounds, not all of them, but many of them, can dissolve in water. It's important to notice that in the solid state, ionic compounds do not conduct electricity well, only when they are dissolved or melted. Now, not in addition to ionic bonds, but another type of bonds are covalent bonds. In covalent bonds, electrons are shared. So, for example, in the molecule of hydrogen or chlorine, these molecules consist of 2 atoms of the same element, 2 atoms of hydrogen, or 2 atoms of chlorine. Essentially, we can illustrate it the following way. So hydrogen and hydrogen, chlorine and chlorine. When atoms are identical, the electrons are equally shared between them, and we call this nonpolar covalent bond. When the atoms are different, the electrons are shared unequally. For example, here in the molecule of hydrogen chloride, electrons are unequally shared, and electrons are shifted towards the chlorine atom. That results in the formation of polar covalent bond. That shift in electron density from one atom to another results in the partial charges, partial positive charges on the atom which electrons are shifted away, and partial negative charges on the atom that electrons are shifted towards. Basically, we can say that in the molecule of hydrogen chloride, the electrons spend more time near chlorine atom rather than near hydrogen atom, which creates partial negative charge on the chlorine and partial positive charge on hydrogen. In this table, you can see the correlation between the bonds and the polarity of the molecule. And it's important to understand that the polarity of the bonds does not necessarily give polarity to a molecule. For instance, the bond between oxygen and hydrogen is polar covalent, and electron density is shifted towards oxygen, so it carries partial negative charge. The molecule itself is formed as an angle, so the positive charges on hydrogen are not equilibrated by the negative charge on the oxygen, which makes the entire molecule polar. In a molecule of methane, every bond is pretty much nonpolar covalent, and the methane is a nonpolar molecule. But what about carbon dioxide? The bond between carbon and oxygen is polar, and we're going to discuss why. But since the molecule is linear, the polarity of the, let's call it a left bond, this one, cancels out the polarity of the right bond. So overall, the molecule is nonpolar. So why is electron density shifted towards a certain atom? This feature of atoms, the feature describing the ability to attract electrons, is called an electronegativity. So more electronegative atoms attract electrons stronger. And essential electronegativity determines how the electrons are distributed between two atoms in the same bond. So in polar covalent bond, the electrons are shifted towards the atom that is more electronegative, and it acquires partial negative charge. The greater the difference in electronegativity, the more polarized the bond is, and the greater the partial charges on the atom. In pure nonpolar covalent bonds, the difference in electronegativity between the atoms is less than 0.4. In polar covalent bonds, the difference in electronegativity lays somewhere between 0.4 and 1.8. For example, if we look at the potential bond between nitrogen and phosphorus, the difference in the electronegativities is 1, so it will represent polar covalent bond. If the difference in electronegativity is more than 1.8, the bond is technically considered ionic, although there are some circumstances when it's not true. For our intents and purposes, as long as you know this distribution, you're good. So electronegativity increases from left to right across the period and increases down the group. We can represent it this way. Electronegativity increases in this direction, and increases in this direction. You can see it on the right. Electronegativity goes up from bottom to the top of the group, and it increases from sodium to chlorine. We don't take into account noble gases because they have complete octet and they're not characterized by electronegativity. So essentially, the greatest electronegativity is concentrated in the upper right corner among nonmetals. The lowest electronegativities are at the bottom here, the elements of the first group, left bottom. And as I mentioned, noble gases aren't included because they tend not to share electrons. Now, the final bit of conversation that we have about bonds is the conversation about polar covalent bonds that facilitate the formation of hydrogen bonds and van der Waals bonds. So imagine you have a polar covalent bond between hydrogen and oxygen. That results in the partially positive charge on the hydrogen and partially negative charge on the oxygen. These partially positive charges on hydrogen and partially negative charges on oxygen form what's called a hydrogen bond, which plays a critical role in biological systems. For example, hydrogen bonds formed between water molecules. Hydrogen bonds are usually weak. They're easily broken, for instance, when water evaporates. But hydrogen bonds is a perfect illustration of the statement, the strength is in numbers. Multiple hydrogen bonds manage to hold the strands in the DNA double helix, and they play an important role in defining the shape of the proteins. Another type of a very weak bond that participates in stabilization of biological molecules is van der Waals interactions or van der Waals bonds. They depend on the fluctuations of electron densities. Essentially, van der Waals bonds formed when electron distribution between the atoms fluctuates temporarily, and that creates temporary dipoles. What is a dipole? But remember, it's temporary. So two dipoles can interact like this, forming weak van der Waals bonds. Van der Waals bonds are the ones that give geckos the ability to crawl upside down vertically on the surfaces that are absolutely smooth, such as the glass. That concludes our first week conversation. Next week, we're going to talk about properties of water and organic molecules.